All right. Um, Welcome back. So this is what I'm gonna do. I'm gonna do a continuation of where we left last time. Life sciences. Hopefully by the end of the day, we will be done with this topic called the chemistry of life so that we can jump on examination examination questions. So without any further ado, let us continue off where we left. Last time, as far as I remember, we covered all of these units. We also discussed the proteins and we did the experiments under the proteins, the lipids, as well as carbohydrates. We did the food test and I gave you the simulation link where you can also perform the experiment by yourself. So remember, we really cannot meet in person where we can do or perform our practicals in person. So we were able to perform our practicals online using the simulation link on the internet. So now, what I'm going to do today is I'm going to continue with the enzymes, nucleic acids, and the vitamins. Hopefully, we will be done with this topic where we're going to end with the vitamins so that we can do the exam questions and jump to another topic for term number 1. Now, let us start with the enzymes. Let us learn what are enzymes. Remember, enzymes also fall under organic compounds, right? And we have learned what are organic compounds as a refreshment, quick reminder or preview. We have learned what are organic compounds. Enzymes um, is one of the substances that falls under the organic compounds. So now, what are enzymes? We need to learn what are the enzymes, right? Now, the enzymes are also called the proteins. It is true we have learned about the proteins, but at this point in time, you need to know that the enzymes are also regarded as proteins. Okay? And therefore, the enzymes are biologically catalysts that speed up the rate of chemical reaction without being used up in the chemical reaction. So now, this is what I love to do when I talk about the enzymes. Let's say, for an example, you want to be friend with someone, right? And that someone also wants to be friend with you, but let's say I am the middle person. So now what I'm gonna do is I'm going to tell your friend that I'm going to tell that someone that you want to be friend with that you told me that you want to be friend with them. And therefore, I'm going to do the same thing. I'm going to tell you that that someone told me to tell you that that someone wants to be friend with you. So now this is how it works. This is how enzymes actually works, okay? Enzymes, they speed up the reaction to take place without being part of that reaction. So that means I am an enzyme. I'm going to tell someone that you want to be friend with them. And I am going to tell you that someone told me they want to be friend with you as well. So that you guys can be friends without wasting time. You guys can be friends without me being part of your friendship. What did I do is I just made the reaction to take place quickly. So that means that's how enzyme actually works. Enzymes intervenes between two substances without being part of that reaction. That's what enzymes do. Okay? So now, the chemical reactions that take place in living cells are known as metabolic reactions. This is not my first time telling you this. The reactions, the chemical reactions that are taking place in the cells, in the living cells, are called the metabolic reactions. It does not matter what type of chemical reaction we are talking about. Photosynthesis, it is a metabolic reaction. Cellular respiration, it is metabolic reaction. So meaning any process that is taking place, any chemical process that is taking place in a living cells are called the metabolic reactions. So enzymes may be involved in two types of metabolic reactions, that is anabolic and catabolic reactions. So that means, remember, I said to you, metabolic reactions are the reactions that are taking place in the cell. So these proteins or these enzymes, they may be involved in metabolic reactions. So the specific metabolic reactions are namely anabolic and catabolic reactions. When you talk about the anabolic reactions, we are talking about the reactions at which we are building up. So when you build something through a chemical reaction, we are saying that reaction, it is anabolic. It's when we build up. But catabolic reaction, it's a type of reaction whereby we are breaking down. So meaning anabolic and catabolic, these are the reactions that are taking place in a living cell. So the other reaction, it is building up. The other reaction, it is breaking down. But generally, these reactions are called the metabolic reactions. So enzymes, which are proteins, may be involved during those chemical reactions. Remember, what are enzymes? They speed up the chemical reaction without being part of that reaction. They make reaction to take place quickly or faster. That's what enzyme do, or that's what enzyme does. Now, during an anabolic reaction, a complex molecule is built up from simple molecule. Remember, I said to you, anabolic reaction most of the time, it's when we build up, like I said. So during anabolic reaction, what is going to happen? A complex molecule is going to be built up from a simple molecule, meaning we're going to take the simple molecule. This simple molecule, they're going to react with one another, or they're going to interact with one another. In these simple molecules, they will be able to build a complex molecule, or to build the complicated molecule. So this is what will be taking place during the anabolic reaction. And do not forget, anabolic reaction, it is a type of metabolic reaction. Anabolic reaction, it is a reaction that is taking place in the cells, in the living cells. Now, during the catabolic reaction, a complex molecule now is broken down into simpler molecule. So that means we're going to take the complex molecule, we're going to chop that complex molecule into the simplest molecule. Meaning here we are simplifying. We're taking something that is complex and we are breaking it down to a simplest molecule. This is breaking down. It's called the catabolic reaction. So the keyword, this is what I do, because sometimes I know you might confuse catabolic reaction and anabolic reaction. So kata, it's like you're cutting down, you're chopping down. So meaning you're breaking down, okay? So that you really do not forget, use this catabolic kata as if you're cutting something down. So do not forget, I repeat, catabolic and anabolic, they are called metabolic. So these reactions, these two types, kata and ana, these two types of reactions, they are taking place where? In the living cell. So remember, the enzymes may be involved during these types of reactions, which is ana and catabolic. Now, during the digestion of food, when you break down food molecules, right? During the digestion of food, complex molecules are broken down into smaller molecules. That's what we know, right? We eat large chunk of food and we start by chewing inside of our mouth using our teeth. So that process of chewing, it is called mastication. It is called mastication, okay? So the first process of you digesting food, it's when you chew, right? You're gonna eat food, meat, whatever, and then you chew. That's the first process of you digesting food. So during the digestion of food, complex molecules are going to be broken down into smaller molecules. And therefore, these reactions cannot take place without the help of enzymes. Like I said, enzymes, they speed up the chemical reactions. So that means when we digest the food, we're going to take the bigger molecules and we're going to chop those bigger molecules, or those complex molecules. We're going to break them down into smaller molecules. But in order for that process to take place, or in order for that reaction to take place, we need the enzymes. The enzymes are the middlemen. They make the reaction to take place faster or quickly. So we need the enzymes so that we can break down the food molecule into smaller molecules. And that is happening during digestion of food. Remember, this is chemistry of life. So I hope that was clear behind the enzymes. I hope that was clear how these enzymes are. What are enzymes? Are proteins. What do enzymes do? They speed up the reaction without being part of the reaction, chemical reaction. And we have learned that we have the metabolic reactions. There are two types of metabolic reactions, anabolic and catabolic. Ana is to build up, kata is to break down. And therefore, we see that for food to be digested, where we're breaking down the complex molecules of food, we need the enzymes. Now, since when we are going deeper in the enzymes, there is a model of enzymes. How does actually enzymes work? Okay, this model, it is called the lock and key model of enzyme action. So here we're gonna learn how does an enzyme actually work? How does an enzyme actually work? So we're gonna see that through the model, like I said, which is called lock and key model. Lock and key model, it explains how enzymes act, how enzymes work, or how enzymes function. So the substance on which an enzyme acts is known as the substrate. So that means we have an enzyme, right? We have what you call the substrate. That means enzyme will work on substrate or will act on substrate. That's where enzyme will start to take action. Without substrate, enzyme wouldn't be able to work because it needs substrate. But you will see what is a substrate. Okay, that's the first thing. Okay, let me jump here. So what we see over here is we have this. It's an enzyme, okay? And then this, it will be the substrate. The green color, it will be our substrate, okay? And then the gray color, it is our enzyme. So this enzyme, it needs substrate so that it can act. That's the first thing that you need to know. The second thing, when enzyme is in action, the substance that is formed during the reaction is the product. So that means here is enzyme interacting with substrate, and therefore this is the end product. This is the final product. Enzyme, after functioning, it will release our substrate as a product. So you can see that the substrate is going to break down. We are breaking the substrate down into two pieces. Now these two pieces now are going to be called the products after enzyme has acted upon a substrate. Enzymes are substrate specific. So what does that mean? That means specific enzyme can only act on a specific substrate. Example, the enzyme sucrase will only react with sucrose, etc. So that means enzymes are specific. Really, enzymes are super specific. You know, let me give you an example that you will easily understand. Let's talk about you know that a washing powder, the soap, the dishwasher, the liquid dishwasher, those are made up of enzymes. The soap that you use to bath, the white this thing, the powdered soap that you use to wash your clothes, the dishwash liquid that you use to wash your dishes, those are different types of soaps, right? But you need to understand that those soaps are made up of enzymes. Now, the reason why we have a soap for dishes is because that soap, it is designed for substrates for dishes. The reason why we have powdered soap is because that powdered soap, it is designed for clothes. You cannot use a liquid dishwasher to wash your clothes. No, because the enzymes are specific. They want the specific substrates. So that means the lock and key model won't be able to function properly because these enzymes are not going to be able to fit directly into the substrate at which it doesn't belong to. So that's why we're saying the enzymes are substrate specific. They want a specific substrate. It is the same as you taking a key of your mom's or your bedroom and wanting to open the main door with the key that you use to open the bedroom. It won't work because that key it is specific for a certain lock. You understand? That's how enzymes work. They are specific. So now the substrate fits into the enzyme's active site as a key fit into a lock. An enzyme substrate complex now will be formed. That's what I have just explained to you. But if I go back to this diagram, like this is an enzyme, right? This is a substrate. And then this part over here that we see over here, let me use my laser so that you can see what part am I actually talking about. This part over here, this part, this section over here, it is actually called, that section that I have showed you over there, it is actually called the active site. The active site, it is where the substrate will be able to fit, because we know that this enzyme, it is specific to this substrate. So that means there is no any other substrate that will be able to fit over here, because other substrates, they're going to come with different shapes, and therefore other substrate wouldn't be able to fit on this active site of this enzyme over here. So that's why we're saying the enzymes are substrate specific, and therefore those substrates, they're going to fit through the site which is called the active site. So that's what we are trying to explain over here by saying the substrate fits into the enzyme's active site as a key fit into a lock, and enzyme substrate complex is formed. Now, when this substrate and this enzyme fit together through this site, which is called the active site, we're gonna end up with something that looks like this. This looks perfectly fine. It fits very well. It can, now the key can work. Now, what do we call this combination? What do we call this combined substrate and enzyme? We call this enzyme substrate complex. It's enzyme substrate complex, whereby enzyme, it is now fitted together with our substrate. We're going to call it the enzyme substrate complex. So now again, okay. Now the reaction takes place and the substrate becomes the product which leaves the enzyme. And therefore now the reaction will take place. And from here we can tell that the enzyme changes shape slightly as substrate binds. You will see that the enzyme will start to change shape a little bit as the substrate fits, right? And therefore here we can see that the colors are starting to change now because the reaction has taken, okay? The reaction has taken place. And therefore in this case, the enzyme's product complex, now we have the enzyme product complex because now the color has started to change. So now we can see that the reaction takes place and the substrate becomes the product which leaves the enzyme. Therefore, the enzyme is now free to react with another substrate molecule. So now we can see that after enzyme has interacted with its substrate, now the substrate will leave now as a product, will no longer leave as a substrate, will now leave as a product to show that the chemical reaction has taken place. And therefore the enzyme will remain with its shape once more again, so that this enzyme can receive or can interact with another substrate. Because remember, enzyme, it is not the part of the reaction. So in this case, we just wanted to make a reaction without being part of it as enzyme. So now we can see that the products leaving active site of enzyme here are now products leaving the active site. Now we still have the active site, and therefore this enzyme, it will be waiting for another substrate that looks like this to come in. Therefore the reaction will take place again, and therefore that substrate will start leaving the enzyme once more again as a product, and so forth and so forth and so forth. And therefore that's how the enzymes actually work. So now, as much as we've learned more about the enzymes, what are the enzymes? What are they used for? How do enzymes actually work? We saw that how do enzymes actually work? We just discussed the log and key model. Now, because enzymes can work, it depends what enzymes are we actually talking about. So now we have effect of temperature on enzyme activity. Like for example, like I said, soaps, dishwasher, liquid dishwasher, the powdered soap. Those soaps, they work under different, like they work in high temperatures. If you want to wash your dishes, you don't use cold water to start with. If you want to wash your body, you really do not use cold water. If you want to wash clothes, apparently we can use cold water, we can use hot water, but normally we use cold water to avoid the stretching of clothes, right? So that's why normally we use cold water. But you need to understand that these enzymes, they function under certain temperatures. Enzymes are specific at which temperatures they function under. So some enzymes, they wouldn't be able to work under high temperatures. Some enzymes wouldn't be able to work under low temperatures. You know, enzymes, they prefer their own specific temperatures. There are enzymes that can work best at high temperatures. There are enzymes that can work best at low temperatures. So it depends. Okay, so that's why we want the effect. That's why you're going to learn the effect of temperature on enzyme activity. So now, enzymes are sensitive to changes in temperature, which is true. Okay, so now most enzymes are inactive. Most enzymes are inactive at low temperatures. So what does an inactive mean? Meaning they do not work. Not that they are dead, not that, no, they do not work. They can't work. They can't perform their function. Meaning that thing of substrate coming to the active site, joining with another enzyme, that won't be able to take place at low temperatures. When we're talking about the cold temperatures, so most enzymes, most, not all of them, most enzymes are inactive at low temperatures. But the temperatures at which enzymes work the best is known as their optimum temperature. Example, they can work best within this range between 35 degrees Celsius and 40 degrees Celsius. This is the optimum temperature at which enzymes can work best. But now when you talk about the enzymes loses its shape and function and denatures at the temperatures above the optimum temperature. So now when you talk about the high temperatures, most of the enzymes or some of the enzymes, they won't be able to function. Remember, enzymes can't function under the low temperatures. Some enzymes cannot function under the high temperatures. So when enzyme it is not able to function under low temperature, we say enzyme becomes inactive. But when an enzyme cannot function under the high temperature, we're going to say the enzyme has denatured. It is denaturing. So what does denature mean? Denaturing meaning is when an enzyme loses its shape and loses its function. You will see this when you're getting rid of. There is an agrosome. It's an enzyme, okay? Agrosome is an enzyme that is used during fertilization. So enzyme, agrosome, it is an enzyme that comes from the male. So we have what you call the testicles, and the testicles are not supposed to experience high temperatures. So when the testicles experience high temperatures, so that means the acrosome, because it's an enzyme, it is going to denature, it is going to lose their shape, it is going to lose their function. And therefore, during sexual reproduction, the formation of a human, the acrosome won't be able to function, and therefore it will lead into fertility. And therefore they will say a male it is infertile. Why the male is infertile? Because his testicles experience the high temperatures. Therefore, high temperatures they lead it to acrosome, which is an enzyme, to denature. So when that acrosome denatures, it is going to lose its shape, it is going to lose its function. Look, this is an enzyme, right? This is an enzyme. So if it loses its shape, this active site, it won't even, this enzyme won't be able to fit there. because now the enzyme has lost its shape because of high temperatures. So the moment enzyme loses its shape, therefore the lock and key model won't be able to take place because the enzyme has lost the shape. So we need this kind of shape so that this thing can fit. Okay? It's the same as maybe when you melt, because we have metal, right? The lock, not the key, the lock, the lock of the key, where you insert the key. If maybe you melt the lock of the key, experience hitting it with high temperatures, it is going to melt a little bit and lose the shape. And therefore, when you try to insert the key, therefore now the shape has been changed. Now you won't be able to get the key inside of the lock. So that's just how it functions, okay, when it denatures. So remember, low temperatures, inactive. High temperatures, denatures. That's how it works. But the enzyme can work best under these temperatures between 30 degrees and 40 degrees. So apparently, um, the temperatures of testicles supposed to be between 2 and 5 or 2 and 3 degrees Celsius. So it's not 35 over. So that's what I'm saying to you. The enzymes are specific on their temperatures. There are enzymes that can work best at low temperatures, even though they say most of enzymes are inactive. But some enzymes, they can work best at low temperatures, while some enzymes can work best between 35 and 40 and so forth. Okay. So now here we have a graphical representation of enzyme versus the activity of enzyme versus the temperature, because we just spoke about the effect of temperature on the enzymes. So now here we have the graph, right? And then this graph, we have the percentage activity against the temperature in degrees Celsius. So now here we have the temperature. So now here we have temperature 0 degrees, 10 degrees. We can see that within this interval, there is no percentage activity of enzyme. That means there are no enzymes that are working here. Here we don't have enzymes that are working. But maybe between 10 and 20 degrees, we can see that from 10 to 20 degrees, which could be somewhere else over here, we have at least 10%, 11, or maybe more 20% of enzymes that can work best at this point in time. But when you have a look at the peak, which is at, we can see that the temperatures above here could be, this is between 30 and 40, right? In the middle right here, we have 35, obvious. So maybe 38 to 40, just like they made that, just like here, oh, here it's 35. So maybe 38, even 35 is the best. You can see that when the temperature is 35 degrees, we have about 90 something percent of enzymes, activity enzyme. So when we have 38 somewhere there, so our enzymes, they're gonna work best. They're gonna work at 100% over there. And therefore, now we can see that starting from here, above these optimum temperatures, we can see that the enzyme will start to lose their shape, right? They'll start to not working properly now because the temperatures are starting. That's why the activity increases, the decreases, sorry. That's why the percentage activity starting from 100 to 90, it drops. You see, 50, 40, 30, 20, 10, and therefore, and so forth. So here, the activity of the percentage activity of the enzyme starts to decrease. Therefore, it leads to enzymes being denatured. I hope that was clear. Now we're going to talk about the effect of pH on enzyme. Not only temperature that affects the enzymes, also the pH does affect the enzymes, the activity of enzymes, or the action of enzymes. Ne? The enzymes maybe that can work under low pH level. Low pH, when you talk about low pH level, we're talking about the acidic level. But when you talk about high pH level, we are talking about the basic level, right? So apparently enzymes are also sensitive to changes in pH. Enzymes has a pH range within which it can function. This is known as the optimum pH. Enzymes denature at extreme pH levels. The extreme pH level is when it is too low, like it's when it is too acidic. The other extreme pH level is when the pH it is too high. meaning it's too basic. When you have too much of a base, when you have too much of an acid, 14, that's too basic. 1, that's too acidic. So those are extreme pH levels. So enzymes, when they experience those extreme pH levels, we say they become denaturing. They denature. We know that by now. What does denature mean? It means they lose their shape. So here again, we have the presentation of the graph, percentage activity against the pH. Once more, again, we know that pH ranges from 0, in this case, to 9 or to 14, basically. So we can see that the optimum temperature is between the optimum pH. It is between, you can see here we have 7 and then here we have 8. Between 7,5 and 7,9, somewhere over there. That's when we're talking about the optimum pH at which enzymes can function best. Okay, so that's how it works. So remember, at the extreme low pH level, acidic, too acidic, enzymes will denature. At extreme high pH levels, such as benign, here we're talking about the basic, this extreme temperature, enzymes will also denature, they will lose their shape. Here, there's no way we can say the enzymes are going to be inactive. The enzymes can be inactive under the temperature, which is what we have learned so far. And therefore, what are enzymes in everyday life? This is what I have just spoke about. Enzymes in everyday life. Where do we use enzymes in everyday life? Like right now, when you stand up and say, you know what, I'm going to use the enzymes, what are you going to do? Now, let's start with biological washing powders. Contain enzymes, example, lipase and the protease that can break down the stains. It's when in this case, our substrate, it is a stain, and therefore our enzyme will act on that substrate, substrate, which is a stain, and therefore the reaction will take place at high temperatures. And therefore, meaning, I get when you're washing your clothes, or let me say, yeah, when you're washing your clothes, it depends on the temperature, right? You can't wash clothes with cold water, right? So when you are washing your clothes, temperatures, it is important. The pH, it is also important. So when you are washing your clothes, washing your clothes, the enzymes from the soap or washing powder, it will act to that stain. That stain, remember, it's your substrate. And therefore, they will fit together, lock and key model, and therefore the product will be released. When the product gets to be released, it's when the stain get out of the clothes. That's how enzymes work in everyday life when you use the washing powders, because they have the enzymes, the washing powders. And don't forget, the enzymes are also regarded as proteins. Again, the enzyme, so if you want to be a chemical engineer, this is what you're going to learn about. The enzyme protease breaks proteins, right, down into smaller soluble molecules that can be removed by cleaning agents in the washing powders. Like, for example, let me say you were eating and then you were eating food that has proteins, and therefore it happens that the food gets into your clothes. What are you going to do? You're going to wash the clothes, right? What are you going to use to wash the clothes? You're going to use the washing powder. And that washing powder has what? Has the enzyme protease, so that this enzyme can break the proteins of the food that you have just ate. And therefore the stain will now be removed from the clothes. So we are done with the enzymes. We don't have any practical experiment under the enzymes. We have learned a lot about the enzymes, and I hope everything was absolutely clear. Okay. Now the big question, what have we learned so far, you, my students? We have learned about the enzymes. Yes, talk to me. What do you understand about this subtopic of enzymes?
What do I understand? That enzymes are proteins. And there are two types of let's say, can I say chemical reactions to enzymes?
Those are metabolic reactions.
Which
Do not
are
forget.
the, yes.
What are
Which
metabolic reactions? Let's start here. What are metabolic reactions? What do you call them, the metabolic reactions?
are they anabolic and catabolic reactions?
Okay, so where is metabolic? I get metabolic, there are two of them, Anna and Kata. So where is metabolic reaction taking place? Where can we find the metabolic reaction? Where is metabolic reaction taking place? Is metabolic reaction taking place in the tissues? Is metabolic reaction taking place in the hair? Where is metabolic reaction taking place?
What are your questions? Askisa Godlo Katil.
Where is metabolic reaction taking place?
I don't know.
In the cells. I said in the cells. That means you didn't focus. The metabolic reactions are taking place in the living cells. Right? So you need to know that. Okay. So
Okay.
so make sure that you go through this and so forth so that we can continue. Sorry?
Okay. I said I will.